here to present the topic potentials of Parkia, biglobosa in agrochemical and allied industries. I believe my presentation will be the shortest because I'm just landing. I just dropped my bag and came in here only to see that I'm meant to present because I thought I'll be presenting in 2 days' time. So I'll just give us what I have. Now, Parkia biglobosa is one of the underutilized spice plants. Normally it's about 7 to 30 meters high. Then that's the inflorescence. Now we have the young fruits that when they are matured and the seeds are broken out, we have the seeds displayed there. Now we have most parts of the plants that are used as food. They're also used for medicinal purposes traditionally. Now we want to see, since they have used traditionally, we can incorporate them into the industries to see how they can help us better. Now distribution, you see that they're distributed from Uganda in the east to Senegal in the west. Now the economic importance of Parkia biglobosa, we have the seeds. The seeds, when they're fermented, normally call it dawa dawa in Yoruba language, has a high protein content, used in cooking soup, stews. Some people add it to sauces. Now that means traditionally they are used as food, added to sauces. They can be incorporated into the food industry. Now the raw seeds can serve as coffee substitute. That means they can be utilized in the beverage industry. Now when the leaves are used, they could be mixed with cereals, and some people eat them raw as vegetables. You can also find them useful in the food industry. Now the fruits could be eaten by man or even livestock. That means they can be incorporated into the feed of animals. Now medicinally, we have the bark, the gum extract. They can be used as toothpaste by the locals. They can be used for treatment of ear complaints, mouthwash. Now you realize that if they are incorporated into the industries, they could be used in making toothpaste. Now we have some toothpaste that are used, some of us, we have bleeding gums. So you have particular toothpaste that you could use for your bleeding gum. You have other ones that you could be using when you have ulcers. Then the macerated bark could be introduced into bathing water to treat burns, to treat hemorrhage. Then the flowers can be used for treatment of hypertension, leprosy. You can find all these useful in the pharmaceutical industries. Now I realize that most of the locals, when they use this, they don't have a dosage. You just go ahead and use them. And some of them might even use them at an overdose percentage, which might even have an adverse effect on them instead of treating what they wanted to use it for. So now, if the pharmaceutical industries or other industries that find them needful, make use of them, you realize that before any drug is being released, we have prescription. So they are going to have a particular dosage for them to be used. Now I realize that most locals also use it as substitute because of the high protein content in the seeds. Now they can find use in industries, and most cereals industries, they can be incorporated into them. Now potential food sources, when we have these foods, realize that most of them are not even enough for the locals. Now if they are incorporated into the industries, realize that at least it will reach us, even those that don't even like the plant itself, because most of them say that the processed products has a little odor that scares people away. So now if they are processed and they are incorporated into the industries, we'll find them more needful. Now what we actually had to do was to check the nutrients and anti-nutrient composition of these plants and see how they could be useful in the allied and agrochemical industries. Now what we did was to process the raw seeds. We had to boil the raw seeds After boiling, we dehull them by pounding in the local way of producing it. They had to pound to dehull them, and after which we fermented in the local way, put them in wooden baskets and covered them with leaves. Then the fermented product, we make them in bowls. Then we now stored them in airtight bottles until they were used for the experiment. Now we collected them from 3 different locations, mostly Northern Cross River. We have Obudu, Banliku, and Buki local government area. Now for the proximate composition, we realized that we had high moisture content in the fermented product most. Then ash content. Realized that ash content depicts mineral content in a particular plant. Now since it had high ash content, especially in the raw product, realized that the plant has potential high mineral content. Now for the crude fiber content, realized that the boiled product were having higher fiber content. I know that any food with high fiber content could help in fighting cancer. That means they can be incorporated into industries to make cancer drugs. Now fat content. We had fat contents were highest in the fermented products. I realized that most high fat content means that it could be used as substitute in foods. I know that fat content also is a prerequisite for making soaps. Then protein content. You know that most of our food, really most of us cannot even be able to afford the animal protein that we have right now. Most of them are not even accessible. Now this could be used as a substitute to the animal protein, meaning that we can incorporate this into foods and we can have the protein that we need. Then carbohydrate content, also see that they were highest in the raw products. Now for the anti-nutrient composition, we realized that the polyphenols, they had high content, though in one of their sessions it was highest in the raw product, but processing in Obudu and Obandiqua session, we had that the polyphenol level was highest in the fermented product. Now the phytic acid content were also high in the raw product, but when they were processed, they were reduced. Then the oxalate content, we saw that they were high. Now very high anti-nutrient composition in plants will make them potentials for incorporation into pharmaceutical industries, because we realize that most of these drugs that are being produced now, some of us cannot even have access to it because they are too costly. So if this is incorporated, we believe that drugs from these products might be at an affordable rate for us to reach. Then amino acid profile. We know that to utilize the protein content in a particular food substance, we look at the amino acid profile. Now from these experiments, this was the profile that we had. Normally people report that glutamic acid is the most abound amino acid in plants. But from this result, we had aspartic acid being the one that gave us the highest result. Now, for us to utilize the protein level, we had to compare our results with the World Health Standard. For the Obudu session, we realized that the results that we had were comparable. The first bar there is what we had from their own standard. So we realized that from our own results, at least each of the essential amino acid that is compared here is comparable, while some of them are even higher, meaning that this serves as a good protein source. Even from the Bukia session, in short, from the 3 sessions we considered, amino acid composition was comparable with the whole standard. So with this, we can see that this can actually be a very good protein source for food. And seeing the anti-nutrient composition in the seeds. We can also have them useful in the pharmaceutical and other industries. Thank you very much.
Any questions? Yes, please.
Thank you, sir. The presenter, thank you for a good presentation. I have particularly enjoyed it. Mostly in the course of your presentation, you were talking about things we can derive from these plants in the future. But you will agree with me that presently in Africa, Nigeria in particular, the main use is as condiments in cooking.
Okay.
Yeah. So, and you mentioned something about eliminating the present flavor or odor that it has to encourage more people to accept it. I would rather advise the opposite. Most people that eat dawa, especially in Nigeria, it's that particular flavor that attracts them. So if it's going to be utilized in the food industry, I think the flavor should remain. Then again, the fat content that you showed, a bit high. What can be done to eliminate this? And what kind of fats would that be?
What can be done to eliminate the fat content?
The fat, because it will definitely discourage people from consuming.
Well, initially it was only the fermented products that were consumed, but now people find need to consume the boiled and the fermented products. Now, if you really need the fat content, that is when you concentrate on the fermented products. But you realize that the fat content was lower in the boiled products than in the fermented products.
On one of the slides you have shown the aspartic acid content and glutamic acid is more, but they are not essential amino acids, no?
Sorry?
In one of the slides you have shown the content of this amino acid content, that is aspartic acid and glutamic acid, very high when compared to the other amino acids. Yes, aspartic acid and glutamic acid.
Okay.
They are not the essential amino acid.
They are not essential.
Amino acids.
No, what I was saying was, people had said that glutamic acid is always the highest in plants. But in our own results, we realized that aspartic acid was even higher than the glutamic that has been reported as the highest amino acid in plants.
What it indicates? What it indicates?
It also indicates the potential use of this plant as a protein source, because the protein content is distributed in the amino acids.
Yes?
Perhaps I missed on this, but what kind of fermentation do you refer to? The fermented product, when you say fermented product?
Anaerobic fermentation.
But do you add special...
No, just the natural...
The natural surface, like natural yeast or...
I did not add anything. What we just did was immediately after boiling, we just transferred them, and just covered them with the leaf. So we left them to ferment naturally.
Yeah, like you did wine in the old days, natural fermentation of the yeasts on the surface. Yeah.
And for how long is that?
Just for 72 hours.